So before I start, I just want to point out that this work that I'll be talking about is done by a group of people, and most of the key people are here on the slide. David, of course, and Brian Daniels, I want to mention, is that ASU has been involved in a lot of this work in particular. So we in the collective computation group, we study how the noisy strategies and decision rules of heterogeneous actors combine to produce emergent function at the aggregate scale, and we work on a variety of different types of systems. That's just a little bit of context. And I want to start with this quote that I believe, David, did you pull this out and put this on the agenda? This quote from Murray that I think is really nice. It's from his Nobel acceptance speech. How can it be that writing down a few simple and elegant formulae like short poems governed by a string of rules, so forth, can predict universal regularities of nature? And the reason I want to start with this is because this is what we do as scientists, of course. But my talk is going to stress that what we need to do better, what we need to be explicit about, is figure out how nature does this. And I don't think they're necessarily the same things, or the answers that nature comes up with and our answers are not necessarily the same. Okay. So why are there apparently so few universals in complex adaptive systems? I think there actually are many, but right now it doesn't seem that that's the case. So given the properties of adaptive systems, perhaps there's a problem in how we identify them. And this is going to be the sort of structure or the content of my talk. I'm not going to tell you about existing universals in information processing systems. I'm going to talk about how we might go about studying information processing systems given their properties to figure out what the universals are. And to do that, I want to really take seriously what nature is doing. Okay, so let me start with some observations. So physical systems are dominated by concepts like pressure, temperature, and entropy. And these emerge through simple collective interactions and provide deep insights into the structure of the universe. And so this is kind of illustrated in this caricature on the slide here. And biology, of course, makes use of comparable collective concepts like metabolism, robustness, conflict management. But in contrast to in physical systems, these are functional concepts. This is a big difference, and it's really important. There's feedback. Now, both physics and biology produce order through the minimization of energy. But adaptive systems produce order with the addition of information processing. It's not to say there's no information. Of course, information plays a role in physics too, but it's incredibly important in biology. So why adaptive systems have this extra step, and whether it makes them fundamentally subjective and uncharacterizable by laws or unamenable to prediction, are big open questions that pretty much, or many of us in this room are interested in, certainly at the Santa Fe Institute. And so I think we need to think seriously about information processing, and we need to think about its implications in biological systems. And to do that, we first have to think about or consider the properties of biological systems. So I put this picture of a cell up here because it's the sort of model of a cell that many of us have in our mind. And we think this is what a cell is. Maybe not if you're working in cell biology, you don't think this, but for the rest of us, it's kind of like what we hold in our heads. And of course, it's a model. It's a model, and it's sort of taken the place of a cell. And that's a danger that I think we need to be very aware of, when our models take the place of the phenomena itself. And the reason why this is dangerous is because this is actually what a cell looks like. This is incredible, right? This blows my mind. So, and it's organized. This stuff is in the cell. It's got all this stuff has got a position in the cell. It's not chaotic. It's highly organized. You can see elements of the previous image in this cell, the mitochondria and the nucleus and so forth. But it's incredibly complicated. If you look at animations in this Smart Biology textbook, I highly recommend you take a look at that, because they've got some beautiful animations. They look like cities. Just like if you watch the documentaries by Godfrey Reggio, when he speeds up the dynamics in cities, looks just like what's going on in these cells. Now, of course, that's one lens for looking at these things, but the world is incredibly complicated all the way down. That's the point I want to sort of make here. And I'm going to keep making it over the next couple slides. So now we have a cell here, this incredibly complicated thing, and we've got a human body, and of course, naively, we often just think of, yeah, we've got some skeleton, we've got some organs, and so forth. But in fact, what's actually going on in there is we've got 30 trillion self cells, 38 trillion bacteria, right? So basically one-to-one self-other. 86 billion of these are brain cells. And there's 8.6 million people in New York City. I mean, that is just think about New York City. What is New York City? So just amazing stuff, right? Now, if you're sort of coming naively into complexity science, and you have this idea that society is at the most complex level, and you look at this and you look at that cell, and then you think about the human body, that seems wrong. But on the other hand, you could say, again, I think just a little slightly more sophisticated way, that actually it's a kind of Russian doll model of complexity, and society is more complicated simply because it contains all the other stuff. That doesn't strike me as very interesting. And so there's a legitimate question here of actually, what is more complicated, and is that even really is maybe the right point that it's all complicated, that we see complexity all the way down. Okay. Now, so what do we have there? We have many body systems with incredible complexity, multiple scales, hierarchically organized, and we also see across biological and other adaptive systems lots of error. And I could give, or I will at some point, give a talk on error in biology. I'm not there yet because it turns out that really we don't know how much error and optimal information processing there is in biology. I haven't seen anyone do a taxonomy or a catalog of all the types of errors or the distribution of errors in biological systems. I think and by error I should say I mean deviation from the maximum theoretical regularity that could be extracted from the environment. But there seem to be a huge number of errors in biological systems, and a lot of them get corrected, right? That's why it's a sort of robustness discussion. I have here just one error called selective attention bias from the human case, where you probably all know this. This comes from Christopher Chabris and Dan Simon's group, where they showed an audience or a group of subjects a video of a group of people playing basketball, and they asked the subjects of the study to count the number of passes between, I think, the people in the white shirts or something like that. And during the video that the subjects were watching, a gorilla runs through the basketball game. And pretty much no one sees it. It's hard to believe, right? I mean, you probably have seen this by now, so maybe you're not so surprised anymore. But nobody sees the gorilla, and it's called a selective attention bias. And the point I want to make here is that this deviation from maximum theoretical regularity extraction in the environment, there are lots of things that give rise to that. Some of those could be good reasons, like trade-offs, like speed accuracy and others. Something like that's going on here. And some of them are just genuine errors in the colloquial sense. But they appear to be rampant in biology. Now, as one caveat to that, we do know that there is occasionally optimal information processing in the work of Bill Bialek and his colleagues at Princeton and elsewhere on segmentation development in fruit fly larvae is one example where the information processing appears to be optimal. Okay. So what do we have in biological systems? We've got many body systems. They're all collective. They're higher order interactions. I won't say much about that today, but it's a big interest of mine and some other people, Josh, in the room. There's complexity. It appears at every scale. The components have partly aligned interests, and they're strategic typically, whether in an evolutionary or learning sense. The systems are largely they're non-equilibrium systems, and they're error prone. Okay, so this error prone thing is a big interest of mine. My question is, to what extent does that introduce subjectivity in information processing? And David and I and others have this idea that adaptive systems overcome subjectivity by collectively computing solutions to environmental challenges in evolutionary learning time. All right? Now, there are lots of examples of the use of the word computation in nature. This is certainly not a new area. And I'm just going to flip through a bunch without explaining any of the details, just to give you a sense of the range of areas in which this has been thought about to some extent. So gene regulatory circuits computing traits and features of the body plan of developing animals. That's probably one of the most canonical examples. This is kind of a newer one that's kind of cool. Heart muscle cells have nanotube circuits that compute the relaxation and contraction of the cell behavior. So they actually have little circuits within the cell made of nanotubes that do this. It's not just by diffusion, the signaling is not just diffusion, which is super cool. These microbial power grids in the soil that shuffle electrons to compute metabolic functions. Of course, neuroscience is this vast area. Neuroscience, neural circuits compute decisions at the whole organism level, like this monkey here trying to decide which way dots on the screen are moving. Social insects like ants and termites adaptively compute architectural properties of nests to match the environment. Temperature, for example, they change the airflow and the density of the internal structure of their chambers. Primates use signaling circuits to encoding the collective vote to compute power structure. So there's tons of examples in biology, but there are lots of problems with these examples. And just to give you a sense, like this work by Eric Davis and his colleagues is incredibly beautiful and rigorous experimental work. They did over many years, and you get these circuits, these gene regulatory circuits, describing how simple features of the sea urchin body plan develop, like the position of cells in the developing layer of the skin. And this particular one is for like 30 hours since, I don't know, what is it, birth or fertilization? I always get that wrong. I should know that. And it's incredibly complicated, right? And so we have all sorts of questions like, well, if you were Darwin, you might ask, well, how is that feature, the developing layer of the skin, why is that important? Why are you focusing on that aspect of phenotype, right? Is that functionally relevant? We could pick any macroscopic property. Why that particular one? If you're Babbage, you might look at this and think, and anyone who's worried about agent-based modeling, this is the quote, "On 2 occasions I've been asked, pray Mr. Babbage, if you put into the machine wrong figures, will the right answers come out?" And he responds, "I'm not able rightly to apprehend the kind of confusion of ideas that could provoke such a question." Boltzmann, don't just make him being but silly here, where's the theory, right? So we have this incredibly rigorous starting point, in a sense, this map that's been made from these experiments of the causal interactions between genes and proteins and so forth. But what's the theory for how the input actually ends up producing the phenotypic output, right? And how can we approach that more rigorously? So if you think about those examples that I flipped through, what's the structure of the problem here? It's not like the physics case. It's not as simple as the physics case. And I don't want to suggest, and I think this is a mistake my colleagues and people in biology often make, that the physical systems are simple. I think they're not actually simple at all. They just look simple because we've worked on them for so long. But there are some differences. And let's see, is this pointer? So in biology, in adaptive systems, and by biology I also include social science, we've got the environment. We need to take that into account. And the big problem, as I said earlier, is one of the big problems is information processing. How do the XIs extract regularities from the environment? All right? And then there's this construction or computation process. How do the strategies that the XIs come up with based on the regularities they've extracted produce the macroscopic properties of the system? Bob was talking about this when he said that biological systems construct their macroscopic worlds. That's my phrase, but he used something very similar. And so we've got some algorithms that describe how the X's get the regularities from the environment, and then we've got some aggregation over those strategies that are a result of the regularities to produce the macroscopic property. And then, of course, there's feedback. And I thought a lot about this feedback process. I'm very interested in it because I think what's really going on here is the X's are making reads. They're reading off these macroscopic properties that they're constructing, and then using the regularities as these things converge or consolidate to make decisions. All right? And I call that effective downward causation. And if I had more time, I'd talk more about that. But the main point for now is that subjectivity that comes from the idiosyncratic information processing means that the right micro and macro variables are not going to be obvious a priori as they might have been in the physics case. We need the system's point of view to figure out what the right macroscopic properties are and what the right input is. And we're going to have to titrate, I argue, between these two to figure that out, to get the natural scales to fall out of our analyses. So I want to treat the micro to macro mapping as a collective computation that's performed by the system in its search for configurations that reduce environmental uncertainty. And so all of the way we think about this is mostly in terms of information theory and uncertainty reduction. And I'll say a little bit about that at the end of the talk. Okay. So collective computation. I want to step back again for a minute and make a point that I alluded to when I introduced the examples I flipped through. Computation is well worked on in biology, of course. We all know that. But for the most part, I would say that the approach, like in the Eric Davidson case, has been what I would call pragmatic, meaning that they do beautiful work to understand the GP map of echinoderms. They're not interested in the foundations of computation. It doesn't undermine the quality of the work in any way. It's just not their question. They're interested in explaining, coming up with theory for GP map. That's great. But so they pragmatically borrow often ideas from computer science like circuits and algorithms and so forth to do a better job modeling biological systems. Another approach is what I might call CS foundational, computer science foundational approach, where biologists or computer scientists or others are actually taking concepts from computer science like Turing and applying them to biological systems or asking if they can. And so a recent example of this is a paper by our colleague Juan Perez. And they showed that actually a simple one-pot chemical reactor can recognize the context-sensitive grammar. And the point they were making there is that I don't want to get into the details of this, is that you don't need sophisticated biochemistry to do complicated computation. So that's a kind of example of a more foundational approach from computer science applied to biology. We don't think that's the right way to go, and we're not the only ones. Walter Fontana and a lot of other people have sort of objected to this wholesale borrowing from computer science. And there are sort of two themes under what I'm going to call the pragmatically foundational approach that we use and that Walter has used to this. And the first is, I'll go with Walter's, is that he wants a formal language. He and his collaborators wanted, when they did this work quite a number of years ago now, a formal language based on mechanism, and they were studying chemical systems, to come up with a theory of computation in adaptive systems. So that's related to Church and Turing, of course, but it's taking seriously how chemical systems work in their case. And the pi calculus is an outcome of that. And you can read Walter's papers on alchemy, also related to that. Another approach is to say, well, before we can even do that, we need to really understand how biological systems work and what the elements of computation in biological systems are, what the architecture is. And then once we have that understanding, then we can think about developing a formal language of computation that's right for biology. It doesn't come from computer science. And we might come back to Turing. I don't think so, but we might. Okay, so the types of questions we could ask along these lines, these are how I see the questions, are questions about capacity. What are the limits of neural computation, for example? What range of expression patterns or phenotypic traits can a gene regulatory network compute? The standard, argument everyone has is the brain a universal computer? Another type of question we might ask concerns satisficing efficiency, optimality. Does the computation efficiently and robustly produce an optimal output? Can the computation be refined over evolutionary or learning time? How does it get sort of like updated? How does the fit to the environment, maybe measured through something like mutual information, improve over evolutionary time? Mechanics and architecture. Is there a logic to the circuit of the computation? There's a lot of work in biology on this. Is it decentralized, or does it have causally influential nodes? Right? Are the contributions additive or synergistic? Josh might say a little bit about this in his remarks in the panel. I don't know. He thinks about some of these issues. Does substrate matter? Finally, evolution. Are the mechanics of some computations more evolvable? Most of what I will say for the remainder of this talk concerns architecture, the architecture and mechanics question, and a little bit of the satisficing optimality. Okay, so the elements of computation, I won't go through this in too much detail, but we're trying to think through what the main elements of computation are in biological systems. And here I've got some circuits from some work we did on primate societies. These circuits, the individuals are nodes. The nodes are individuals or pairs of individuals in the C12 and 21 cases. And the edges link individuals who have strategies for each other based on the decision rules they use to join fights. So monkeys have big fights. In these groups, the fights can vary from like 2 to 30 animals, actually, in a group of on the order of 50. They're sequential, typically. So David and I have a fight, and David redirects to Jen, and so forth. Jen redirects to Josh, and I drop out. So you get this sort of complicated fight, and then there's a sequence of these over the course of an observation period. And you can ask, how do individuals make decisions to join fights? And it turns out in these groups that those decisions are predicated to some extent on who was fighting in the previous fight. And so you can extract decision rules from a time series or strategies from a time series, and that are causal rules the individuals use for deciding to join a fight. And we can use those rules to build circuits like this. And then the circuits, of course, individuals have multiple strategies. So the individuals have to decide if they have a conflicting strategy, like David was in the fight previous to me, and Jen was in the fight previous to me. And when Jen's in the fight, I fight. And when David's in the fight, I don't fight. So now we have these two conflicting rules, and we have to combine them in some way. So there are gates that specify how the rules get combined so the individual can produce a behavior as a result of the strategies that it has developed. And you can think about these strategies, and I'll bring this up again later, as coarse-grained representations of their fight history, coarse-grained representations of what they've learned about who interacts with whom and what the outcomes of that are. Okay, so these are circuits that we build from the time series data. And like I said, the edges the weights on the edges represent the probability an individuals play a particular strategy. So here, and more generally, the input is a stochastic strategy of components. And don't worry about the boundary stuff. It's a little too complicated for today. And the output is going to be a functional macroscopic property. Here, that's the distribution of fight sizes. And the reason that's important is because larger fights cost more. So it's a functional macroscopic property. And then there's some algorithm or circuit that describes how the strategies combine to produce the output. In this particular case, the algorithm is not so important, but in other work we've done, the algorithm aggregates all of the votes of individuals or all of the strategies to produce some output, and it turns out to be very important what algorithm is used to do that aggregation. And what we're interested in is how the system identifies a good algorithm for aggregating information. I'll come back to that at the end. And then there's of course, and it's very important to have a way to make the program stop. And so you might think when you're thinking about a biological system, well, here's a very obvious area where computer science ideas don't apply at all, but I don't think that's true. So instead of a termination criterion, we have a notion of correctness. And the notion of correctness is basically, there are different ways to think about it, but we've been thinking about it in terms of slow variables and timescales. So the system creates a hierarchy of timescales, and the slow variables provide background against which the system can tune or make decisions. So it becomes a kind of effective, the background becomes an effective, gives them effective notion of correctness. And so it doesn't tell the program to stop, but it gives you a sense of what's the right or good response, coming back to the optimality question that I had a couple slides ago. So there's no assumption that there is an optimal or correct computation. It's developing as the system is constructing those macroscopic variables. The correctness is, of course, I've just said this in a number of different ways, operationally defined from the point of view of the system. And we'll come back to this at the end. We sort of operationalize that in terms of mutual information. But it is good to know as a scientist whether there's a kind of optimal computation. And the reason for that is it gives us a sense of how good the system is at figuring out what the environmental regularities are and whether the answers or solutions it's producing are maximizing mutual information with the environment. Okay, so what's our abstraction here of what's going on? So I have here, this is a sort of the collective computation computer, so to speak, that we've been developing. It's just a schematic of it, and it hides a lot of complexity. Here we have the W's, and the W's are the perceivable states of the world, and they can be some exogenous ground truth, like the height of the Eiffel Tower or the amount of oxygen in the soil, or they can be endogenous ground truths, meaning that they're constructed by the system dynamics collectively. So that would be like what happens during an election when we come to a collective opinion about who's a good president. It doesn't always work so well. But that's more of an endogenous one. And so the exogenous ground truth, the important point there is that this is something that's effectively outside the control of the system, whereas the endogenous one is usually being built by the system, but then takes on a slow time scale and becomes a kind of internal ground truth. So the axis here is a circuit, and it's composed of opinions or strategies of the sensors or individuals. So the circuits that I showed you in the previous slide will be in here. And the X's are basically to build those circuits. The X's have some algorithm, P, that's going to map the state of the world onto their behavior. All right? And then we have the Y's, and the Y's are simpler, complex algorithmic aggregations of X. So basically the Y's are the macroscopic properties in the system, and the A is the algorithm, the aggregation mechanism that takes all the votes or decision rules of the X's in the circuit, the important information in the circuit, and combines them to produce the macroscopic variables. Now, the individuals do something with this information. So in the systems that we've studied, the individuals can not only make estimates of these, but as these consolidate and become more reliable, the individuals start to make estimates of the macroscopic world. So this comes back to Bob's point, and it's about how systems figure out what, how systems make estimates of semi-global or global variables. And then there's a translation B from their interpretation of the macroscopic world to behavior Z. Okay, how much time? A few more minutes, David? What? Rough? Okay. So I have just a few more slides and really quick on principles that come out of this. The first is information accumulation and information aggregation. So information accumulation, often called evidence accumulation in neuroscience, concerns how the X's build their effective theories for how the world works. I just said that. And the information aggregation are the algorithms by which the strategies of X's combine to produce the Y's. And there's some interesting, you can trade off aggregation versus accumulation. Let's say you know that there are a lot of biases in how the X's interpret the world. So you build a better aggregation mechanism to compensate for those biases. Okay. There's the principle of collective encoding, which just says that the information, this is an important one because the information is encoded in the network, not in the heads of individuals or of the components. So it's in the interactions, the principle of collective encoding. And it doesn't mean, of course, materially it has to be in the brains or the processing components, processing parts of the components, but the information is really collectively encoded in the network or in the circuit. And then there's this principle of agency, and I think this is a really important one that's actually not appreciated enough in computer science. the principle of agency says, so in the cases that I've been describing, the x's are reading off, as I said, these macroscopic variables. And so it's that reading that makes this a computation and not just a computer. A computer is a device for performing a computation. A computation has some agency. The actors have to be making estimates or reading off information in order to perform the computation. And this was a point that I've made in this paper on coarse graining, and was made nicely by Clara Horstman and her colleagues. Walter found this paper a couple of years ago for physical systems. And I think this is my final 2 slides. So the principle of coarse graining uncertainty reduction. So as I said earlier, the x's have this algorithm for estimating the w's, and they're essentially making coarse-grained estimates of how the world works. And this is important because it comes back to some points in Bob's talk. The principle of coarse-graining uncertainty reduction, I think, changes how we view complex systems once you start looking at the world this way. What we have is not so much simplicity and then emergent complexity, bird flocks and so forth. What we have is complexity at the microscopic scale and complexity at the macroscopic scale. And we have some coarse graining for the purposes of uncertainty reduction by components. They're making models of the world. Those models are making an information bottleneck at the mesoscale. And then this allows a macroscopic expansion, allows novel behavior to emerge at the macro scale. All right, so my model of how complex systems work is not simplicity to complexity, although it's sort of in here if you're sophisticated about it. It's complexity, information bottleneck, through coarse graining, macroscopic expansion. Okay. So let me just finish up by saying that one of the things we're concerned with is, are there predictable relationships among macroscopic variables? Are these relationships fundamental? That means for me, can they be derived from the microscopic, thermodynamics, statistical mechanics? And if so, then we can say that the relationships are law-like. And collective computation is a theory for how the macro is constructed from the micro in information processing systems. Okay, so you can't do it in the physics way. You have to take into account how the system's computing, how it's coming up, how it's estimating regularities, how it's coming up with its own effective theories for how the world works. And so my final slide is just this. We'll observe law-like regularities in collective adaptive systems when the collectives build effective theories for how the world works that reduce uncertainty. And these effective theories can capture ground truths or can result in the construction of a macroscopic world that captures largely regularities in the collective dynamics. That's the end. Thank you. Yeah?
Yeah. Would you make a bubble about how this bottleneck idea relates to modern machine learning algorithms?
Well, so what I will say is that, so the term information bottleneck, I use endogenous coarse graining. The term information bottleneck I borrowed from Tishby. Tishby actually has a paper on what he calls may not technically be identical, but he calls the information bottleneck, and he uses that to describe some of the layers in our artificial neural nets that do compression. And so there is definitely a connection, but I'm not ready to say precisely what it is.
Okay. Well, just to follow up, there's a general principle of information theory that the general decoding problem can't have a bottleneck. It can only have a bottleneck, which is to say a rendering of what's happening, if the signal you're trying to process has some special properties. And so there's a question upstream of the design question here, I think, which is how does that restriction relate to the problem you're solving?
So I'm not sure, but I think a related point is, so when we do coarse graining, of course, it's lossy. And, but at the same time, so we're losing information, but at the same time, the components are gaining a better understanding of what the right strategy is. So in some sense, they've gained information. And that's what allows this information expansion, the microscopic expansion I mentioned, and the novel behavior. All right, so we don't really have a good way to talk about this yet. So you get this coarse graining that allows them to figure out what the regularities are and make better decisions. And that is more informative for the space of strategies that they choose among. So it's interesting. I think it's related to your point, but I'm not sure it's identical. Yeah, Melanie.
So I was interested in your idea about errors in biology and notion of optimal computation, because it seems to me that that's very context dependent. What counts as an error and what counts as optimality? So like the gorilla in the basketball game example, selective attention, the task was to count, and there were lots of regularities in the environment, including the gorilla, that should be ignored. Yeah. So what's your thought on that?
Yeah, so that's why I define, so I'm very operational about this, that's why I defined error as the maximum deviation from the maximum theoretical regularity extraction in the environment. Right, so that's my definition of error, and anything that deviates from that is an error. But some errors are for good reasons, and some errors are just because the system doesn't work well. So the good reasons are speed accuracy trade-offs, selective attention bias, and so forth. When you have multiple things that you need to keep track of, and you can't, you don't have full attention for all of them, right? There's lots of reasons like that. But I want to keep error clean and keep it tied to the maximum regularity that you could extract from the environment. And then, come up with a taxonomy of reasons why you might get that deviation. And I think this maybe will come up in the panel in some of your remarks, I hope, because this is a big issue in neuroscience, of course, like how we make models of the world and so forth. Yeah. So any other questions? Some of Pablo and Mercedes.
Jessica, thank you very much. One question is, would you predict that this computation and this kind of mutual information between the collective and the environment will increase somehow with the group size? Will be linear as cumulative evolution of a culture people will think, or there will be a non-linear function to the extent that there might be a threshold size beyond which you get a lot of noise and you don't make sense of the environment anymore. And the other related is there any scope for variability? Just thinking of the human phenomenon, I mean, we are collective that we somehow decode the environment in different ways. So there might be some, maybe there is a maximization of mutual entropy, but there's many ways to do that somehow.
Yeah. So the first question, I think, the answer there is it's probably some nonlinear function. But what I think happens is that we innovate, and we biological systems, human systems, develop new mechanisms for reducing the variance as group size goes up. And one of the questions that I'm interested in is, when does this work and when doesn't it work? What are the conditions? And so I think even with increasing group size, you can, you still can get this optimal or semi-optimal solutions for information processing, but you have to invent new mechanisms for control. And the second question, I think, and you tell me if I get this right, but one of the things we're interested in is when do the estimates, the coarse grainings that the individuals or the components of the systems are making converge? When do they start to have similar estimates of the environmental regularities? What are the conditions under which that happens? And the reason why that's important is because as their estimates converge, that's going to mean that the macroscopic properties that are being constructed through their interactions are going to be easier to read, because they're going to have less variance too, right? So obvious answers to that are when they have similar computational capacity and similar exposure to the environment. But of course, it comes back to the error point. We know that in biological systems, components often see only a part of the world, and the whole reason for being collective is to combine all the little pieces that we see into the whole picture. So, I don't know.
Yeah, maybe this is the last question. Sorry, Mercedes, you go, and then we'll have a 10-minute break, just because we're off time.
Maybe a trivial question. Can you say something about collective computation and thinking about levels of selection if we are thinking about systems like ecosystems? And sorry for the question. And the other is, I find the slow time that you talked about in correctness really interesting. And I think in a fascinating way, like asking how the processes at the micro scale, the same forces that are creating your whatever information bottleneck, etc., may be set in that time scale. And I think that in the context of diversity and some things I study, that is really interesting. Like what sets that
the time scale?
The slow time. Yes, the slow time scale.
So with respect to the first question, I mean, going way back to even my undergraduate years, one of the motivations for this work was deep frustration with levels of selection. It's just not mechanistic, right? It's so when we do this work, part of our reason for doing this work is to figure out a way to identify the natural scales of the system without imposing them. And then once we have an idea how the scales arise, that gives us a sort of a better understanding of how to model the evolutionary dynamics that sort of stabilize that. So there's definitely a relationship to the levels of selection thing, but we think the right way to do it is to do it from the microscopic first. And the second question, the slow variable question, so the coarse-grained variables are slow variables, sort of by definition, and we study how these coarse-grained variables arise through interactions and component models of the world. And one of the things that what we think is that the value of these coarse-grained variables is that they give the components something to tune against, right? So they stabilize or provide this notion of correctness, essentially. And so it's not what sets the. I can give you an example. So what sets the time scale, and so one of the monkey systems that we work on, the individuals have fights. And then there's a long history of fighting, and one individual learns it's likely to lose, and if the asymmetry is big, it has a special signal it emits to indicate this, that it's accepted the subordinate role because it knows it's going to lose. And this signal is a placeholder. It's a coarse-grained representation of the fight history. And so I believe that biological systems make mechanisms like this signaling process that allow for these nice slow variables to reliably arise. And so I think when you ask what sets the slow variables, we have to look for mechanisms that summarize things, essentially. All right.